I wish you a very warm welcome to this Top Research Seminar on personalized medicine, organized by the LUMC-wide research theme Medical Genomics. So almost a year ago, when this seminar was planned, I naively thought the seminar would be in person again. Well, instead, we are again at the start of a fresh lockdown in a light version. And our clinical colleagues are in the middle of yet another wave of COVID patients. And I can imagine the postponement of planned care that can't really wait. Well, back to business. First, a few words about our research theme. So we are here to connect, support, and inspire LUMC researchers who study human health and disease through the lens of the genome. We have organized ourselves into 8 sub-themes: genomic therapeutics, pharmacogenomics, single-cell genomics, epigenomics, tumor genomics, clinical genetics, functional genomics, and population genomics. Every theme has an ambassador, and you can contact them if you want to know more about the sub-themes. Today, your hosts are the management team of the management team members of our theme, Gijs Santen, Sylvie Noordermeer, and myself, Bas Heijmans. The guiding principle of our theme is that we are about the collective. So each member has influence and impact. So, and if you want to know more, because you are not yet a member of our theme, but you are interested in medical genomics, you can scan this link to sign up, or you can contact us at medicalgenomics@lumc.nl. Today, the topic will be personalized medicine. That's a theme that's very much at the core of medical genomics, but also at the core of many of the activities within our hospital. First, we will have 4 state-of-the-art presentations of the research going on within the LUMC on this topic. Next, we will have an external speaker who will look forward with us what will be the impact of personalized medicine on our healthcare system if it all works out. And we have an interactive discussion or on the question whether the LUMC is ready for the implementation of personalized medicine. But first, I would like to ask to the stage Willeke van Roon-Mom, who will present on the possibilities of RNA therapy in personalized treatment of neurological disorders. Willeke?
Yes, can you see my screen?
Yes.
So thank you, Bas, for the introduction. So I'm gonna update you on strategies for personalized treatment for brain disorders using RNA therapy. So it really all started with this landmark paper of Mila Sen, where there was a little girl with Betten's disease who 1 year after her genetic diagnosis received a personalized treatment for her unique DNA variant that causes Betten's disease. And when Annemieke Aartsma and I read about this paper or heard about this little girl, we said, we have all the expertise in-house within the LUMC to replicate this. So we could also apply this for Dutch patients. So how did this work? Mila was diagnosed quite already when her disease was quite advanced, and she received her treatment for a few years. So unfortunately, she died at the beginning of this year, but her parents said that over the years of her treatment, it was really an enormous improvement in her quality of life, and that is sort of illustrated here in this slide. So Mila suffered from quite severe epileptic seizures. And so the color of the dots indicates the length of the seizure. So the darker the dot, the longer the seizure, and the higher the dot, the more seizures per day she had. So you can see that over a period of 2 days, there was a really significant decrease in the number and the length of seizures that Mila had during the day. And this for her was really important for an improved quality of life. So we thought we can do this, and it is really for a very unique set of patients. So what if a patient has a texture that is caused by a variant that is so unique that there are less than sort of around 5 patients worldwide? And industry will never be interested in developing therapies for such low number of patients. And it's also not possible to perform classical clinical trials on this group of unique patients. And in Europe, it's not necessary if you want to develop an RNA therapy for these small groups to register them at the European medicine agencies. So at the same time as Annemieke Aantsma and I said we can do this, we will start the Leiden Center of RNA Therapeutics. Rob Collin and Radboud UMC, who is also developing these RNA therapies, but then for genetic eye disorders. And Ipe Elgesma at Erasmus, who is working on neurodevelopmental disorders. They were all thinking the same thing. So we thought better unite our forces and start the Dutch Center of RNA Therapeutics. And I think it's really important to stress that this is really a multidisciplinary approach. So all the way from the clinic to the pharmacy to the basic research labs, we have designed these therapies for larger groups of patients, but we know how to design them, make them, and treat the patient. So it's really a team effort. So what is an antisense oligonucleotide? So they're small stretches of RNA or DNA that are chemically modified, and they can bind to the RNA, and thereby they will induce either RNA breakdown or they will modulate RNA splicing. And it's really the type of chemical modification that will determine the mechanism. So the first type of DNA variants that we will focus on in the DCRT are the deep intronic mutations. So what could happen with such a deep intronic mutation is that part of the intron will be introduced in the mRNA, and the mRNA will be out of frame, and the result will be that there is no protein expressed. And then we can design an antisense oligonucleotide that binds this mutation, and it will restore the normal splicing and then also the normal protein expression. So why are we focusing in the DCRT or eye and brain? Because there we can give a local treatment. That means that with a low dose, we will get a high local exposure of the antisense oligonucleotide, and the systemic exposure is low. So liver and kidney damage is not as big a risk. A low treatment efficacy frequency because we deliver locally. And these splice modulating antisense oligonucleotides, they make use of chemistries that are already approved by FDA and EMA for other neurological disorders. So which patients are eligible? The mutations must be very rare, close to unique. Exon skipping must restore the normal protein function. The organ that is affected should be brain or eye. The patient must have benefit of treatment. If a patient has lost all of its sight, then treatment will not be of any benefit. There is no function that can be maintained or rescued. And patients must be willing to undergo this experimental treatment because it will always be an experimental treatment. So when we started this, when we said we can do this, we didn't really know how we were going to do this. In the year and a half, we've discovered that we do not need regulatory approval. So the EMA does not need to register this as a drug. We can apply this under a named patient setting or artsenverklaring. We do communicate with the regulators for advice on how to devise proper toxin safety studies. The hospital pharmacy can prepare the GMP or maybe the GMP-like compound. We try to do the preclinical and safety studies as much as possible in-house to reduce cost. And for now, the funding is done through the UMCs, but in future, we are already talking with Zorg Instituut Nederland to see if we can reimburse the development cost. So this is really the preliminary pipeline that we have envision. So we can in-house design our antisense oligonucleotide and test them in patient-derived cells. We can study if there is protein or function restoration. We are trying to do most of the in vitro and in vivo safety studies in-house together with the animal facility here at the LUMC. And then we can move forward to treatment and deposit sequences. We try to coordinate worldwide to see if there are any other patients with the same mutation that could benefit. There are many initiatives worldwide. The regulatory landscape is quite different. In the US, quite a few patients have already been treated. In Europe, it's quite different. And then within the European countries, it's quite different. So we are trying to coordinate the efforts so we don't have to invent the wheel all the time. So I think the main challenge is that we need to develop these therapies fast because the diseases are quite severe and quite progressive, but we still want to be safe. What is a unique mutation? Is it one? Or if 5 patients have this mutation, is it still unique? Maybe it's 50 patients. No one can tell us what the upper limit is. What is safe? Is in vitro evidence enough for safety? Do we need to do rodent studies? How many rodent studies do we need to do? If we do rodent studies, can we do it under good laboratory practice or does it have to be compliant studies? This is an enormous difference in cost and production of these antisense oligonucleotide. Can we do a GMP or is GMP-like allowed? And we are really trying to align all the procedures and efforts within Europe, but also worldwide. So I think in the LUMC, we are studying an RNA therapy for unique mutations in the WDR45 gene that causes BPAN, which is a neurodegeneration that is characterized by iron accumulation within the basal ganglia. And we do that together with our colleagues in Tübingen. And we need help because I think most of the diagnostics is done by looking for DNA variants in the exons, while these intronic deep mutations are usually missed. So if you know of any deep intronic variants that cause eye of brain disorders, please feel free to contact us. That's it.
Thank you, Willeke, for this beautiful short story. Are there, so Sylvie and Gijs, are there any questions right now from the chat?
No, I don't see anything yet, but maybe people need some time to type in their question. So please, if you have a question, please type in your question in the chat, and then I can raise the question to Willeke.
So what I found striking, Willeke, is that what you try to do is develop treatments for patients, but the number is really small, and yet you seem to need to go to all these regulatory hoops as if you're organizing a clinical trial for Alzheimer's disease.
Yeah, and so I think the main thing is that we don't need to go through all of these safety and regulatory hoops. So the CPG and the EMA said you don't need to do anything. But then when you talk to a neurologist who is responsible for this patient, and the neurologist will say, I think this patient could benefit from a personalized RNA treatment. So then the neurologist is then also responsible. So what and so for someone who's working in the lab, we can design an antisense oligonucleotide, but is it then safe? And that is something that we are now trying to figure out. What do we all agree on is enough proof that this antisense oligonucleotide is safe? And we don't have to do anything. But I think everyone agrees that we would want to have some type of evidence that we are quite sure that it's a safe molecule that we will be injecting in this patient. So there is nothing there. There are some guidelines from the FDA, but yes.
So there's time for a short question. Sylvie?
There is a question from the chat from Christian. In case of patient with eye disease, if both eyes are affected by the disease equally, would you treat one with AON and have the second as a control, or how would you approach?
Yeah, so that is so if we would treat one and use the other one as a control, then that is deemed research. And so that is not allowed under this named patient. So no, we are not allowed to do this, we think. But this is all new. We do not, so you could do this in a clinical trial, but we are not. This is the named patient is different, and the first discussions that people in Radboud, because they're spearheading the eye program, and the discussions they had is that they are not allowed to just treat one eye and then use the other one as a control.
Is there someone at the IRB here or at the LUMC who can support this kind of research/treatment? Is there expertise?
As far as the regulatory and the ethical issues, and we are setting up a network now also, so not just for the Netherlands, I think, because in Germany it falls under the criminal law. So in Germany the regulations are quite different. So we want to figure this out European-wide and then come up with one set of rules that we all need to abide by.
Okay, well, thank you very much, Willeke. We may get back to this in the general discussion. So now I'd like to invite Jesse Swen from the Department of Clinical Pharmacy and Toxicology to share his screen and open his microphone. Yes, so Jesse will tell you about the latest in pharmacogenomics in our hospital.
Yes, can everyone hear me? I think so.
Yes.
Hello everyone. Great to speak here today, and in my presentation I indeed want to highlight some of our works in improving drug response by using pharmacogenomics. We all know that drug response is highly variable. When you treat a patient with a normal dose of a drug, for many it will work, for some it will not work, and some may even develop side effects that can be severe. And for this, obviously, there are many reasons, all of which are studied in the field of clinical pharmacology. But for several decades now, we are increasingly beginning to understand how genetic variation also plays an important role in this variation in drug response. And in pharmacogenomics, we aim to understand how genetic variation influences this drug response, obviously with the ultimate aim to tailor the treatment to the individual patient needs. So when we start a drug, we first do a DNA test, and then based on the test result, we select the dose, or maybe even select a completely different treatment. So yeah, why is this important in the clinic? Well, I could talk for 20 hours on that, but I only have a very short time, so I want to highlight 3 clinical examples where pharmacogenomics would have saved a lot of patient burden without going into too much detail. But the first example is about a very young baby who dies at 13 days, and then in a post-mortem analysis, it was found that he had toxic morphine levels. And these were found to be the result of his mother taking codeine, which is a drug that in the body is converted into morphine by CYP2D6, which is a genetically polymorphic enzyme, and approximately 3% of the Caucasians has a genetically determined very high CYP2D6 activity. So the mother converted much more of the codeine into morphine, and that was transferred into the child through the breast milk. A second example is a male patient who experienced 4 strokes, and following the first stroke, he was treated with clopidogrel to prevent future strokes, and the clopidogrel was continued actually the entire time, and only after the 4th stroke, someone thought, well, let's genotype this patient because clopidogrel is a prodrug that needs to be converted into its active metabolite by CYP2C19. And it was found out that this patient was a CYP2C19 intermediate metabolizer, which is around 30% of the Caucasians. So he could not activate the drug as well as the other 17% of the population can, and he should have received a higher dose or even a different treatment. The third example is about someone who is diagnosed with esophageal cancer, and then very rapidly after starting treatment with capecitabine dies of the treatment, not of the tumor, but she dies of the treatment. And this could have been very well prevented by a pharmacogenetic test. And for this particular pharmacogenetic test, our group has done a lot of work, so I want to go a little bit into more detail. So when you look at capecitabine and 5-FU, they are a large part of those drugs is usually detoxified by an enzyme called DPD. We know that 3 to 5% of the population is deficient for that enzyme. So as a result, they cannot detoxify the drug, and they're exposed to much higher levels of the drug and the active metabolites, and they have a much higher risk for severe, potentially fatal toxicity. So for quite some years, it was known that genetic variation could be an important cause for this DPD deficiency. And together with the Erasmus Medical Center and the NKI, we designed a large prospective study where we tested all the participants before they started treatment with capecitabine or 5-FU for 4 variants in DPYD, which is the gene that encodes the DPD enzyme, for 4 variants which are the main cause of DPD deficiency in Caucasians. And this work was actually performed by Linda Hendricks, who is currently training to become a clinical chemist in the LUMC. So here you see the results of this study. It was a prospective multi-center study in 17 Dutch hospitals. 1,181 patients participated. All were tested for these 4 genetic variants in DPYD. And based on the presence of the variant, you would either receive a 50% dose reduction or a 25% dose reduction. Out of the 1,181 patients, 8% were a carrier of one of these variants, and they received an adjusted dose. And here you see the relative risks for severe toxicity from historical controls. And now in the green, you see the risks for the patients who received a dose reduction. And you can see that overall, this was a positive study. So by using this test, it's possible to reduce the toxicity caused by capecitabine and 5-FU, while maintaining, of course, the efficacy of the treatment. As a result of this study, all Dutch hospitals have implemented this test, and maybe more importantly, also the EMA now recommends this test, so it's included in the drug label, and the EMA recommends testing for all patients who start with capecitabine or 5-FU. So I think this really is a poster child example of translational research going from the discovery of the genetic variants towards implementation into clinical practice. This is only one example of what's currently possible. There is, in fact, much more possible, and that's depicted on this slide. So I already said that for many years we know that genetic variation affects the way how we respond to drugs. And in 2005, we initiated a multidisciplinary working group who reviews available evidence in literature for certain gene-drug pairs. And this is a multidisciplinary working group involving pharmacists, but also medical doctors, clinical pharmacologists, clinical chemists. And we perform systematic literature reviews, and once we find that there is enough evidence for a certain drug gene pair, we write a very specific recommendation how you should adjust the therapy based on the genetic information of the patient. And we have now reviewed more than 100 drug gene pairs, And as you can see on this slide, the drugs are really from all different sorts of clinical fields. And the way the system works is that if you know the pharmacogenetic profile of a patient, so for example, you know that someone is a CYP26 poor metabolizer, you record it in your prescribing or dispensing system. And as soon as you then prescribe or dispense a drug for which we have created a recommendation, then you get a pop-up message on your screen telling you exactly how to adjust the dose. So this system, in fact, is operational in almost all Dutch prescribing and dispensing systems. The only thing that is lacking most of the times is that patients do not know their pharmacogenetics profile. This is available. And this is also very important because pharmacogenetic variation is not rare. So most of the times when we discuss genetics, we think of rare events. But we found out that when you test at the Dutch population or any Caucasian population for a panel of 8 genes for which we have those recommendations in our guidelines available, then you will find out that 95% of the patients has at least one actionable pharmacogenetic variant, and maybe more importantly, 10% has 4 or more. And when you then look at the drugs for which we have recommendations, we also did that. We collected all prescription information in the Netherlands from 2016, and we then calculated that would we have known the pharmacogenetic profile of all Dutch inhabitants in 2016, we would have adjusted 192,000 first prescriptions in that year based on the genetic information. So there is really a lot of probably a lot to be gained by more routinely collecting these pharmacogenetic profiles. So the question then is, are we there yet? No, I can say that we are definitely not there yet. So there are some limitations to our current pharmacogenetic testing. So I'm quite optimistic, so I'm already happy with what we have. But if you were a bit more pessimistic, you could say that by using 0.01% of the variability in the genome, we can now explain 50% of the variability in pharmacokinetics. So there is obviously room for improvement there. And I think we are only beginning to understand the genetic complexity and to account for the genetic complexity in pharmacogenomics. There is ongoing debate on the definition of drug metabolizer phenotypes. We are looking into something that is called phenoconversion, where we know that also drug-drug interactions affect our drug metabolizer phenotypes, and also inflammation has an effect on the activity of some of the drug metabolizing enzymes. And of course, implementation remains an ongoing challenge. Now, very briefly, because I'm running out of time, what are we doing in this area? So we have focused in a large project on the missing heritability of CYP26. And with that I mean that we know from twin studies that 90% of CYP26 activity is genetic. But with our current pharmacogenetic CYP26 testing, we can only explain 39% of the variability. So there is a huge gap there. And together with the Leiden Genome Technology Center, we saw an opportunity when they obtained the PacBio long read sequencer, because that allows the full sequencing of CYP26 in one single run. And we combined that with neural network predictions of CYP26 activity on a continuous scale, while it's normally predicted in categories. We don't have time to go into detail again, but what we did is we took data from the Centrum study, which was a study of breast cancer patients that were treated with tamoxifen, and from all those patients, DNA was collected, and there was extensive blood sampling, and in that blood the endoxifen and desmethyltamoxifen ratio was determined, which is a proxy for CYP2D6 activity. And of all these patients, we sequenced the DNA on the PacBio long-read sequencing, and we trained a neural network to predict the CYP2D6 activity for each of these individual patients. And from that, we learned that by using these advanced techniques, without the techniques, we were able to explain 54% of the variability in CYP2D6 activity, that the neural network was actually able to predict 79, which is a very strong increase, of course. And this was replicated in an independent cohort of almost 200 patients, also treated with tamoxifen, and there the explained variability increased from 35 to 66%, so also a similar increase. And I think this highlights how by applying more novel techniques, we can really again improve the way we can use pharmacogenetics to improve drug response. So I think in this short presentation, I've shown you that there is evidence for single drug gene pairs, that we have created these guidelines that assist in pharmacogenetic result interpretation and implementation, that genetic variation, pharmacogenetic variation in particular, is very common and not rare, that pharmacogenomics is being implemented in clinical practice. However, that there are, of course, are also several challenges, such as the missing heritability, the genetic complexity, and particularly that we have a need for large cohorts with well-defined drug response phenotypes. And with that, I would like to thank you for your attention.
Thank you, Jesse. It was a great presentation. In view of time, I think we should move to the next presentation. And Jesse, could you please go to the chat? There are a few questions. Could you answer them in the chat?
Yeah, sure.
Thanks. So, and then we go to our next state-of-the-art presentation by Maaike Vreeswijk from the Department of Human Genetics on personalized risk prediction and treatment of cancer.
Thank you, Bas. So hopefully you'll hear me well. So this presentation will be about personalized risk prediction, and just start right away showing you the pedigree of Jane, who was diagnosed with breast cancer as early as age of 32. And because also her mother was diagnosed with breast cancer at early age, and both her aunt and her cousin were diagnosed, she was wondering whether her cancer could be hereditary. And if you look to all breast cancer cases, approximately 14,000 in the Netherlands, you see that approximately 25% of all breast cancer patients have indeed a positive family history for breast cancer. And in a subset of the patients, pathogenic germline variants are identified in the 5 common breast cancer susceptibility genes, which can be subdivided according to their risk in high-risk genes that have a relative risk of more than 4 times the population risk. And these are BRCA1, BRCA2, and PALB2, and 2 moderate risk genes, ATM and CHEK2, with relative risks between 2 and 4-fold. So I will focus on the high-risk genes, BRCA1, BRCA2, and PALB2. So why do you want to know if you are carrier of a germline pathogenic variant? This is because you have not only a very high risk for breast cancer, but maybe also for ovarian cancer. And just assume that Jane is carrying a BRCA1 pathogenic variant. She might also wonder about her ovarian cancer risk and might decide, for example, to remove her ovaries because of the high risk of ovarian cancer. Secondly, we nowadays have very specific treatment for BRCA1 deficient cancers called the so-called PARP inhibitors. So she might indeed be treated with these kind of drugs. And thirdly, very important, it's also important to look into the healthy family members because they might be a risk as well. So the DNA test will reveal whether they also carry the pathogenic variant, for example, her brother or her sister. So Jane decides after consultation of the clinical geneticist to do a DNA test. And the outcome of a DNA test can be divided in like 3 flavors. The first group, and that's the majority of the families, I have to say, a benign variant is identified or nothing and no sequence variation at all. So these benign variants, they do not impact the function of the protein. They are just natural variation. And in these families, the individuals are counseled on basis of the family history and also lifestyle factors. And it's important to note that at that case, all women are at risk because you cannot identify specific people that are at risk. This is different when you identify a pathogenic variant in the family. And from these pathogenic variants, these are variants that clearly lead to a loss of protein function. And those are variants like introducing nonsense variants of large deletions and insertions that lead to truncated proteins. And so then you have a totally different situation, which I just explained. However, there's also a third flavor, and these are called the variant of uncertain significance. And from the DNA alterations, you cannot deduce whether it affects the function of the protein. And so most of the time, these are missense variants or variants that might interfere with RNA splicing. And just as in the case of the benign variants, we cannot say whether it will lead to an increased risk or not. So these are only screened on basis of their family history. But it's important to note that these kind of outcome of the DNA test that are communicated with the patients have quite some severe psychological impact because of the uncertainty regarding the cancer risk. And you need to be almost a biologist to understand why a certain DNA variant would increase your risk of cancer almost 6-fold, whereas another variant will not. So it's very important because these VUSs might be benign or pathogenic, and this is not really a small problem because only in the Netherlands already we have more than 800 VUS in BRCA1, BRCA2, and PALB2 over more than 2,000 families, and worldwide there are more than 8,000 of these VUSs. So we have some tools that we can use to determine whether a VUS is benign or not. We look to the prevalence of the variant in cases and controls. We look to co-segregation because, of course, if you have a variant that will be causal to the disease, you expect that all the affected family members carry the pathogenic variant or the variant. And also, of course, we see specific combinations of tumors, for example, in a BRCA1 deficient background. But I have to say that only in a limited number of cases we can show that either a variant is benign or pathogenic. And this is mainly because many of the VUSs are unique and we don't have enough families to do this kind of analysis. So in most cases, VUS are rare, families are informative, and we are stuck with the clinical diagnosis of a VUS. So it's all about the question whether this specific DNA variant will affect protein function. Because as I said, if you have really a loss of function variant, that will be associated with a high cancer risk, whereas if it does not affect the function, it's probably a benign variant. So what is the function of these 3 proteins? They have a very important role in the repair of DNA double-strand breaks via homologous recombination. And when a DNA double-strand break is induced, that is very toxic for the cells because you can lose genetic information that's located at this site. And a cell in collaboration with BRCA1, BRCA2, and PALB2 will copy the missing genetic information from a homologous DNA sequence. And just this will lead to error-free repair. And if you have a defect in homologous recombination because, of course, of some BRCA1 or 2 deficiency or PALB2, you have genomic instability, reduced viability, and also sensitivity to certain drugs like cisplatin or these PARP inhibitors. So we have set up in the Department of Human Genetics with really a nice collaboration between different colleagues a generic assay to determine the effect of the variants on protein function. So what we do is we have these mouse embryonic stem cells and we knock out the gene of interest. So this can be BRCA1, 2 or PALB2. And after that we transfect these cells with the human cell, a human gene that contains either a benign or pathogenic variant or a VUS, and then we measure the functionality of the protein. So of course, if you want to develop a test, you have to validate this test, and this is what we did with many benign variants and also many pathogenic variants, and looked what the functional outcome would be. And as expected, we saw that the viability of the cells was not affected in the benign variants, whereas it was clearly affected if you introduce the pathogenic variant. And also, if you look specifically to the repair of these DNA double-strand breaks, you could see that these benign variants could repair the DNA double-strand breaks to the same extent as the wild type, and whereas the pathogenic variant could not. And the same is true for the sensitivity to these drugs. So in this way, we had a very specific test with high sensitivity and specificity, and especially for the repair of the DNA double-strand breaks, it was very quantitative. So this allowed us to test the impact of these VUSs on protein function. And due to lack of time, I cannot show. We have, I think, almost like 100, 200 VUSs in all the different genes have already been tested. But just to show you how an example looks is if you have this specific VUS and you don't see any effect on the viability of the cells, you see normal repair of the DNA double-strand breaks, and you don't see any sensitivity to the drug, or at least to a similar extent as in the benign variant, you're almost sure that this VUS will not affect the function of the protein. So this means that in all the cases where you do not have enough information to classify the variant either as benign or pathogenic, the addition of functional data will be essential. And so this is what we now do. So we have functional tests to either classify variants as benign and pathogenic. And of course, this is very nice if you can do this in a research setting. But of course, we like to do translational work. So we also want to use it for clinical diagnosis. And this is when we received support from the KWF for a large consortium grant entitled Cancer Risk Assessment Through Functional Testing, or CRAFT. And this is exactly what we do. So if you now look to the pipeline, if a patient has a DNA test and gets a diagnosis of the outcome of the DNA test as a VUS in one of these three genes, these VUSs take like a detour to the functional test street. And then here in the research building, we do the functional analysis. And then we ask all the DNA laboratories, but also the clinical geneticists for clinical data that can be merged for the variant classification for families with this specific variant. And then together in a multidisciplinary meeting with the clinical geneticist and the people from the DNA laboratories, we classify those variants as either benign or pathogenic. And of course, very importantly, we go back to the clinical geneticist, and the clinical geneticist will go back to the patient. So very nice for germline pathogenic variants or germline VUSs, I have to say. But we do not only identify these in germline. We also identify these variants nowadays in the tumor DNA, because nowadays many tumors are being sequenced to identify druggable targets, and we also identify VUSs there. So also for this kind of analysis, it's very important that you can test whether a VUS will affect the function of the protein. And so therefore, when DNA sequencing is not enough to determine the clinical relevance, you need to have functional testing. So with this, I would like to end. I'm very grateful to the Department of Human Genetics, where we can do all this research dedicated to the functional testing of these VUSs. Also supervised by Harry Vrieling, Heike van Atticum and Peter Devley. Also, of course, to the clinical geneticists and all the laboratories in the Netherlands and all our international and national collaborations. So thank you very much for your attention, and happy to take a question if there's still time. So I will unshare my screen now.
Thank you, Maaike. It's really fascinating to see how clinical genetics is developing, and that you now need to know functional genomics to give a good advice to a patient. It's well, a tsunami of work and thinking that needs to be done. I think indeed, as you thought it may be more efficient, I saw that Jesse replied to questions in the chat, which work very well. So I would invite you to also go to the chat and switch off your video, yeah, and then continue in the meantime to the next speaker, Marjolein Kriek. Thank you for being here from the Department of Clinical Genetics on dealing with incidental findings from personalized medical genomics approaches.
Thank you, Bas. I would like to tell you something about dealing with incidental findings within a diagnostic setting, I have to say. But first of all, I would like to tell you something about what a clinical geneticist does on a daily practice. Maaike already told you something about it, but might not be familiar for you all. Well, we talk to patients and we ask them about their medical history, and our organ is more or less the family history. And depending on the reason for our counseling, we also perform some physical examination. And sometimes there is a patient for which you think this could no other be than this specific genetic disorder. And if so, we ask for a dedicated test. However, in a majority of cases, we actually think it might be genetic, but we don't have a specific genetic disorder where we think of. And then we apply for broad genetic testing. And that is whole exome sequencing. And whole exome sequencing is that part of the human genome that's actually coding. And it's 20,000 genes approximately, but still only 1% of our genome. So the patient will come into a hospital and we tell them about the different options within a pretest counseling. And if all parties agree, we draw some blood and that is sent to the genetic laboratory in the LUMC, and together with genome scan, exome sequencing is performed. And then the data will be sent back to the LDGA, and they will perform a very difficult pipeline with all kinds of steps to be able to detect the variant which could be of interest to a patient. If you perform trio analysis, so you have a WES analysis, but we often do trio analysis. What is trio? Well, you need two things for that: the availability of both parents and that both parents are actually not affected. So the trio consists of 2 unaffected parents and the patient. And then more or less we ask the computer, give me all the variants for which both parents are a carrier and transfer them both to their affected child. This is well, the same as recessive disorder. Another thing is, what we also ask is, give me all the variants that are actually absent in the parents but are present in the patient. And of course, we look for X-linked variants as well. Well, I'm very glad Maaike already told you much about the different outcomes possibilities, because of course you could also identify a variant of unknown significance. And what we aim for is pathogenic variants, because then you can tell something about the cause of a certain disorder of a patient, and you can tell them something about recurrent risk and all things like that. But during the pretest counseling, what we also discuss with patients or their parents, depending on the age of the patient involved, is the likelihood of identifying incidental findings. And incidental findings is more or less an answer to a question that has not been raised. So there is a reason why we ask for this test, and we don't get an answer for the reasons why it's tested, but we do find something that is of medical interest to the patient. How big is such a chance? Well, you could imagine if you filter a lot, if you use the parents for filtering, then a lot of autosomal dominant variants that are, for example, related to oncogenetic disorders are filtered out. Same holds true for cardiogenetic disorders. So the chance is about less than 1%. But I can imagine if you, within a research setting, if you look for a specific gene or new variants related to a specific phenotype, and you don't have the availability of the parents, or one of the parents is affected, then you have a higher chance of identifying a incidental finding. So what we do within the diagnostic setting is that we reduce the possibility of finding an incidental finding to the minimum. We don't actually search for findings that are not the reason why we ask for the test. And why not? Because it's not allowed within the Netherlands. If there is no indication for a specific test, then it's called screening. And for screening, you need a license. And that is all written in onze wet bevolkingsonderzoek. And why have we such a stringent regulation? And that is that you have, as a child, you have a right to an open future. So that is our legislation. This is a very busy slide, and again, it's also in Dutch. But what I would like to tell you is that in collaboration with the 7 UMC's within the Netherlands, we have a working group that contains both lab specialists from the clinical genetics department and the clinical, and so the clinical clinicians are involved. And what we try to do is set up a guideline for returning these incidental findings on a national basis. And what we start off with that, first of all, the variants that are pathogenic or likely pathogenic will be considered returning. But if so, a fuss will not be returned. And our first policy rule is incidental findings regarding a clinical feature that can be influenced by a medical intervention. So I can offer something to the patient or their parents for their child, then it will always be reported back unless there is an opt-out. I will give you an example in a minute, but the opt-out is only possible when the age of the patient is older than 12. Within or 12 years or less, there is no opt-out. So, for example, you have an 8-year-old child who suffers from a neurological problem, which is not the same as intellectual disability because he has a normal development and she has congenital heart disease. And what was identified during the trio analysis was a pathogenic variant in a gene that is related to thickening of the heart. We call it hypertrophic cardiomyopathy. And if you have such a condition, there is an indication for periodic cardiac screening from the age of 10 years onwards, and you have a therapeutic intervention if there is a reason to apply this. And furthermore, the father has the same pathogenic variant. So if we identify, or the lab doesn't identify this variant or this condition will be discussed within our committee for incidental findings, and that's for a diagnostic setting. The members of this committee and now the light here goes out, the members are 3 clinical geneticists, 3 lab specialists, the responsible counselor, and the legal and ethical expert. And their role is an advisory role. So we give them more or less an advice how to deal with this incidental finding, but the counselor is actually the one who is legally responsible for everything that is translated back or discussed back to the parents or the patient. So he or she will say, thank you very much, I will follow this advice, or he or she will say, thank you, I will take this into consideration. And if there are arguments just to say this is not what I'm going to tell them, that is the call of the counselor. So the advice here was to report, and well, this is reported back because there is something to gain with this knowledge to the patient, and in this case also the parents, or at least the father. Policy rule number 2 is incidental findings regarding a condition that cannot be influenced by a medical intervention will not be reported back unless there is an opt-in. And as long as the patient is a minor, there will not be a opt-in. But you could imagine if there is, you have a grown-up who insists on knowing everything about his medical condition when it comes to genetic disorders, you could discuss, as long as he understands what the consequences would be, that there could be results back, giving him a higher chance of a neurodegenerative disorder. If he understands it and still wants to have his results back, then that's possible. If you translate it towards a research setting, for example, if you search for new genes for a specific phenotype, then there are several issues which you can consider. First is that participants often want to know everything that has been found within their genome. So either it's treatable or untreatable. They're very interesting to know because they say there's no other way I can know this kind of information about myself. But I could also imagine that if you are doing this research and you identify an incidental finding, so a variant in a gene that you are not familiar with, it's very hard just to translate it back to a clinical setting as being a researcher. So you could say, okay, the alternative will be we deal with anonymous data, but that's also a very, has its own drawback. Just an advice, if you are struggling with things like this, you want to set up a broad genetic testing, but you are thinking of what to do with incidental findings, go to the CMA. They know a lot about this, and especially Dr. Eickholt. She's more or less specialized within that, about this topic. And there will be a wetzeggenschap lichaamsmateriaal who is more or less saying something about returning incidental finding, but that's not ready yet. And the last slide is all the people who are working or have been working on the guidelines of returning incidental findings. And that was my presentation. Thank you very much. I will stop sharing.
Thank you very much, Marjolein. It was a very clear and interesting story. Again, in view of time, I think it would be great if you could answer the question and see one of Jesse, and there may some follow, answer them in the chat, and then we continue during the panel discussion, because I think there's some food for thought there.
All right.
Thank you very much. So I'd like to invite our external speaker, Dr. Robert Vonk, to the floor. There he is. So many of us don't know him. So he's a senior advisor at the Raad voor Volksgezondheid and Samenleving, the Council of Public Health and Society. I don't know if I say that correctly. How I found him initially is that he wrote a report for the Ministry of Health on personalized medicine, together with colleagues at the RIVM, where he worked until 2020. But the main reason for having him today is that he is someone with a very broad and critical view of the history, the present and the future of the way we organize our healthcare system. And I think he will present a very different perspective for us biomedical scientists that's really nice to chew on for a while. So thanks for being
my best.
And please share your screen and let us know what you have to say.
I think it's shared now, right? So thank you for inviting me, and thank you for having me. I must say I was a bit surprised when Bas approached me, and a bit flattered as well. I'm only human. The task he set me out to do is a bit daunting and at the moment, I'm not sure if it's my fault, but I see all the screens in my screen. So I'm not seeing my own, there it is. I'm seeing my own slides now, that's better. A bit daunting because I had to talk about something I did work on a long time ago. when I worked at the National Institute for Public Health, I did some work on personalized medicine, but that's almost 5 years ago. And science progresses. So I'm glad I'm not having to talk or needing to talk about the current state of affairs of personalized medicine, because that's something I don't know anything about. But I am going to talk about what personalized medicine can mean for our healthcare system. And just to make some introductory remarks, I'm not an enemy of personalized medicine. I'm going to point out some impossibilities, which might or might not be impossibilities from the viewpoint of a candid friend. So sometimes you need someone to tell you maybe it's not going to work to set you on the right track. And what I'm going to talk about today is my own personal viewpoint. The Council does not have an official viewpoint on personalized medicine. We do have a viewpoint on personalized healthcare. We're a strong supporter of personalized healthcare and patient-centered care, but not on the personalized medicine we're talking about today. And Bas asked me to reflect on what if, the question, what if personalized medicine delivers on all its promises, what would it mean? That's a broad and very daunting task, because it takes a lot of assumptions to get to that that viewpoint, because you have to assume that personalized medicine has overcome already existing barriers. And I think Willeke already pointed out there are, as of yet, a lot of barriers to fully implement personalized healthcare. So we assume that all translational problems are solved and personalized medicine has become practice changing, and that there are no legal, ethical, and financial issues that pose an obstruction to provide personalized medicine. So those are the two main assumptions behind the story I'm going to tell today. And what would the impact then be? Well, the impact would, of course, be enormous. The previous speakers already pointed out the enormous benefits for individual healthcare that personalized medicine offers. So, yes, the impact would be enormous, would be beneficial, but it will also create, at least in from my point of view, new impossibilities or new dilemmas we have to consider. First of all, what if personalized medicine does all the things it promises us to do? How would we then define health and illness? And how would we organize healthcare? And how would we finally organize solidarity as the bedrock of modern healthcare and accessible healthcare? So these impossibilities are quite profound, and I would like to know what you think about them, about those impossibilities. Do I see things wrongly? Is my viewpoint not correct? We can have a discussion about that later. But first, let me tell you about what I think might happen. First of all, personalized medicine. I took this quote. I just cherry-picked it. It's not. It's something I came across when I was looking for a definition of personalized medicine, and this is personalized before medicine. But I underlined some things I think are very profound. It's about biology. It's biological complexity. It's predictive, preventive. It's personalized, and what I thought was quite. Not really funny, but remarkable is that participatory was put in quotation marks. But what the most striking was is that the promise of at least the authors of this article thought the promise of personalized medicine was that it would force medicine to, or enable medicine to leave the hospital and expand into the homes, workplaces, and eventually schools, and you could say all of society. And that, of course, has its repercussions. because while it's hard to oppose more targeted therapies, for example, we already heard the beneficial health outcomes and how it prevents undesired effects. It becomes a bit different when we talk about predictive and preventive medicine, because we're still talking about risks and predispositions. So we're not talking about people actually being ill or actually having an illness. So in theory, it might work if we take personalized medicine to the next step and also apply it to preventive medicine, predictive preventive medicine. But we should consider that we might be asking too much of people. Not only because health is a multidimensional concept, it means different things to different people, it means different things to different patients. But more importantly, genetic risk or risk in general. And I think Maaike already pointed that out in her talk. Risk is a very hard concept to understand for us, but also for patients, let alone to act upon risk. What are we going to do when I know I am at risk or I have a certain gene or a genetic defect? What am I supposed to do about it? And it's still a risk. So it's and we already see this in normal prevention work. It's hard to invest. It's hard for people to invest in something that might yield returns in the distant future. It might happen. It might not happen. What I'm going to do today might affect that risk or might not affect that risk, and which makes it very hard for individuals to invest into their own health or their own preventive steps they have to take. Furthermore, you almost never have just one disease. It's many patients have many different diseases all at the same time. So what you do have to do as a patient can add up quite quickly. And it might also mean different things and might also mean that you have to do contradictory things. And what are we going to do then? So it really has a profound impact on the definition of health and the definition of illness and the definition of perceived health and perceived illness. But it also has an impact on the way we organize healthcare. And of course, we are in Leiden and some people are at the LUMC. I'm not that far from the LUMC, so I thought I might take Herman Boerhaave, Leiden's very own medical hero, as a case in point, as something that someone who really proposed personalized healthcare, of course, in the 18th century meaning of the word, which is one doctor, one patient, one treatment, which is unique and tailor-made for that patient. On the other hand, on the other part of the screen, you see Jeremy Bentham, which is another side of healthcare. Jeremy Bentham, a utilitarian philosopher, Englishman, has become famous for the quotation that we should strive to a world where we provide the greatest good for the greatest number of people. And it's that utilitarian thought or that thought that is really the bedrock of the current healthcare system. And to explain that, the greatest good for the greatest number of people, I'm going to say something which is becoming increasingly unpopular to say, but I'm going to say it anyway. Standardization has been a boon for healthcare. Without standardization, we wouldn't have the mass healthcare we have today. We wouldn't have a healthcare system that is able to provide healthcare to as much people as we are able to treat today. And in many ways, if we take personalized medicines to its extreme, personalized medicine is the antithesis of modern healthcare. It's tailor-made. It's not standardized. It's tailor-made for the individual patient. And modern healthcare systems, and especially hospitals, are already highly complex systems, which are increasingly unable to provide unique treatment pathways or to facilitate treatment pathways. Remember, hospitals could only become the institutions we know today because they standardized a lot of logistic, financial, personnel, but also medical processes. So providing every patient with a unique treatment pathway is, within the current setting of healthcare, within the current setting of hospitals, actually utterly impossible. It would require more complex processes, more complex financing systems, and a lot more personnel than we can provide today. So what's the catch of personalized medicine in this sense is that what we might gain in health outcomes and patient satisfaction because of more tailored treatments, we might lose in terms of equitable access and affordability. And that brings me to my final point about solidarity. And I can't deny my background. I'm from Zeeland. I was raised in a religious family, so I know my Bible. And I thought this quotation from Ecclesiastes is very to the point. For in much wisdom is much grief, and he who increases knowledge increases sorrow. And that's especially the case for solidarity, because solidarity as a foundation of equitable and accessible healthcare, solidarity cannot exist in a place where we know all the risks involved. We cannot, it's very hard to create a system based on solidarity if we know all the risks involved. And that means that personalized and predictive medicine, the more personalized and predictive medicine becomes, the smaller the basis for solidarity is. And that almost naturally leads into the first irony of insurance, because this is not something that is unique for healthcare. It's also unique for example, all other systems which deal with risks, is that the more you know about the risk profiles involved in what you're doing, the more differentiation you're going to apply. It would eventually lead to unpooling, which means that for healthcare, for example, we tend to split the insurance pool or the financial pool into smaller and smaller segments, which would eventually lead into everybody paying their own way or nobody paying at all. Because we already see, and we already, if you read papers, you already see it happening, people are increasingly unwilling to pay for known risks of others. We already see it in the current debate about lifestyle and health expenditure. Why should I pay for someone who gets lung cancer because he has been smoking all his life? And that kind of debate. We will see that kind of debate happening as well if we know more and more about the genetic risk profiles of people. So personalized medicine is not without its risks, and we can talk about that more later on, of course. But you might ask, Robert, is there still hope then? Of course. To quote another Englishman, hope springs eternal. Because in a way, the impossibilities I talked about are intrinsic to healthcare. They're not really unique to personalized medicine, but personalized medicine does highlight some very special parts or very specific points of those impossibilities. And why impossibilities might be impossible to solve, you can work around them, but it requires radical new ways of thinking about how we organize healthcare. It should really be outside of the box. And it takes a lot of time. Changing things takes a lot of time. So we have to be patient. It will not be done tomorrow and might not even be done in 10 years. But if we want to create a health system that really promotes or sustains or facilitates personalized medicine, we can do it, but we have to be radical and we have to be patient. And I think that's my last slide. Bas, yes, that is my last slide. So I give the floor back to you.
Thank you very much, Robert. It was very enjoyable and also scary. And indeed, I was thinking, so is personalized medicine a thing in China? So, or is this really sort of a Western thing where we sort of put the individual on a pedestal?
I wouldn't really know.
Yeah, I also see some compliments in the chat. Sylvie, is there anything to report from the chat, or shall we go to the?
No, there is a question, and I think it's a nice question from Jan Verschuren, who says, you can argue that smoking is a choice while genetic predisposition is not. Would that make a difference in solidarity?
I don't think so. Well, it must, of course, it makes a difference morally, but the whole point about solidarity, it's easy to create solidarity when you don't know anything about yourself or about others, because you're willing and able to accept things that might not necessarily be beneficial for you, but might be beneficial for the collective. So it's about the knowing and not about the cause of the risk involved. It's about knowing the risks, which makes it hard, because your risk might not be my risk. And it doesn't really matter to me if it's your fault or not. I'm not going to pay for it. That's the way most people tend to think. If they know everything, right? So that's of course a big assumption as well.
Okay, thank you. That was the question in the chat. So maybe now we can go
for a statement.
Exactly. We made a few statements as the organizing committee that it would be nice if you all can vote for what you think. Obviously, there's no right or wrong answer, and maybe the person involved in this statement can actually comment a little bit on the outcome. So let's give it a few seconds to fill in for everyone.
So you can find it in the chat. And people found it. Well, 25. I think it seemed we reached some stability in the answers. Can you maybe also already start, Sylvie?
Yeah, indeed, the numbers are still adding up, but we seem to have a common view that about 30% says personalized medicine is beneficial for the individual, but destructive for the collective, while 70% is not in agreement with this statement.
So is there, Robert, because I think you were clearly worried that it was destructive.
True, yeah.
And
so I'm kind of curious what
yeah.
what the reasons are behind the optimism. Yeah. Maybe I'm seeing things wrong, so it's a possibility.
Maybe I see the Marjolein. Maybe is it because
yeah, I voted no as well, because I was trying to put it in the chat, but then I thought maybe for the discussion it's better to do it this way. Well, you could also argue that, for example, if you do tumor-based therapeutics or pharmacogenetic-based adjustment of your medicine, that being a patient, your therapy is sooner active, or so you could also argue that the healthcare costs involved will be decreased, right? So that would give then all the money that you don't have to spend on me as being a patient, you can give to my neighbor who will also get sick, for example. So there are issues related to personalized medicine for which is actually health cost decreasing.
True, but I don't think healthcare expenditure is the main issue, because I also see the benefits of less treatment, less adverse reactions, better health outcomes. I'm just worried about, are we still able to create a system, accessible and equitable system, once you know all the risks, all the personal risks involved in creating such a system? Because, and I take it to its extremity, right? So it's a bit of a black and white story. We're not talking about ability to pay, but we're talking about willingness to pay. Are we willing to pay for people with a certain genetic defect, of which we know that they will need healthcare, when I, as a person, don't have that risk? So it's not about the expenditure; it's about the willingness to pay.
Yeah, but
that's the main point. So.
Yeah. But I would think that the risk of getting ill is more or less a Gaussian kromme. So the chance of becoming ill will be more or less comparable. And of course, you have the outliers, but that will mainly be based on monogenic disorders, which are very rare. So I would say that the willingness to pay is that the chance of becoming, yeah.
If I can interrupt you, that's very rude. Sorry, but. We know that.
I get your point.
But
also, I was interested by, you said, so now we're talking about this solidarity. So if we throw everything onto prevention, then you may get into this issue. And I thought, well, precision medicine may be fine, but you say, well, maybe not, because we are so successful, because we standardize. And I was thinking of this clinics now that do only one treatment, something with hips or with eyes, and they do this in a sort of a mass production way, and that works very well. So I was wondering if anyone in the audience, are you doing sort of cost effectiveness calculations beforehand when you're designing? I see Maaike, who's here as well. My argument was, we save money if we have more, if we do not ineffectively treat. But your solution was quite high tech, so to screen for the VUSs and whatsoever. I mean, that must cost an enormous amount of money.
Yeah. So I mean,
do you know that we will save money, or do you hope that we save money?
Oh, so yeah. So this is maybe difficult, because of course this is still research funded. So I don't have any cost calculations, but I think that there are many examples. I could have also give a talk about another project that I'm working on, where I think that instead of the one size fits all, we can already at the start of the treatment say, oh, 50% of these people will not respond probably to the treatment. So why give a $100,000 treatment per year on this patient? So I also see it like as well. You started with Swen, was it? I think with we would like to give everybody a treatment that will not do harm, but would be effective. And I don't know. Is that I think that there is an enormous amount of money spent because of the adverse side effects, for example.
And yeah, I was just thinking that maybe also to some extent you can, let's say, standardize individual treatments, right? And there's, I'm thinking of a colleague of us in the pharmacy who's working on 3D printing of tablets, for example, so that you can print exactly the dose that a patient needs. And you let's say, so the doses, the treatment is very tailored, but the way you create the tailored treatment is very standardized. So there, I'm not sure if this.
Yeah, that makes sense, and I think that's one of the ways of making it work, right? So because, and again, I've put it in a very black and white scenario, but if you give everyone individual, a unique treatment, you cannot organize it. It would be totally impossible given the amount of patients we have to process in the entire healthcare system on a daily basis. It's just not possible. But if you find ways to standardize elements of a more individualized treatment, that already would make it more feasible. So it's a way of, and that's what I was trying to do. It's a way of making you think about some of the aspects, organizational, financial, solidarity aspects of personalized medicine. So I think it's a great solution.
Yeah, I think it's actually what you raised in your is sort of this very broad, indeed, the very broad definition of personalized medicine, the 4 Ps. So, and that is, I mean, if you look at it at that, it's really daunting. But maybe if we would think much more about specific situations where you can apply it, and then also think about how you can apply it in a meaningful, cost-effective, organizable way, then it may be possible. So I'm afraid, for example, talking as a biologist who likes new technologies, I mean, if you let me going, I will endlessly trying to find more specific ways of predicting risk or treating diseases, but it may not be useful or possible to implement. And are you, Jesse, for example, you are very far in your work and it looks really good, and also, I mean, you can't object almost by so doing this test and getting the right dose of a drug. Is there anything being done there on how difficult it is to organize, or what the cost benefits or additional costs are of such an approach?
Well, I already in the chat, of course, there were questions concerning the cost, because it comes at a price, of course, to run this test. So yeah, that remains a concern. And then for the DPD test that I showed, it's very clear that it's cost effective. But for a more panel-based approach, those data are not yet there. So there is more research to be done there. Yeah.
There's a question from Annemieke Aartsma-Rus. So I hope it's not only about solidarity, but also about compassion when treating rare disease patients. Yeah.
True. I hope so too. But solidarity is a very technical way to organize compassion, right? On a population level, because the entire idea is that we carry each other's burdens, right? Draagt elkanders lasten. It can get very technical, but in its basic core, solidarity is a way to organize compassion. But it's a very hard way, of organizing compassion is very hard to do. And that's the point I was trying to make. But yeah, I hope so too, that compassion is still there, and that we still feel the need to help those less fortunate in society. So you get no disagreement of me on that question? No, absolutely not.
Well, maybe that should be the final words about compassion, because that's maybe the cornerstone of healthcare and what we would like to see in our society in general. So it's 6:30, well, 1 minute before. So I thank you very much, all speakers, for your great talks and the participation in the discussion, speaking and also on the chat. Thank you all for listening, for postponing your dinner, or maybe sitting with your bord op schoot as if it's Studio Sport. Hope to see you around in a few weeks or a few months, whatever, in the hospital. And thank you very much, and goodbye.